Hello friends, welcome to BMH Learning. We are going to discuss about beta oxidation. Beta oxidation is a metabolic process involving multiple steps by which fatty acid molecules are broken down to produce energy. More specifically, in beta oxidation we have fatty acids. These fatty acids are first converted into acyl-CoA chains in the cytoplasm. This has been already discussed in the first part of this lecture. The longer acyl-CoA chains are progressively converted into smaller fatty acyl-CoA chains. This reaction releases acetyl-CoA, FADH2, and NADH. All of these three then enter another metabolic process called citric acid cycle or Krebs cycle, in which ATP is produced to be used as energy. Beta oxidation goes on until the acyl-CoA chain has been completely broken down. In eukaryotic cells, beta oxidation takes place in the mitochondria, whereas in prokaryotic cells, it happens in cytosol. Now let's discuss the chemistry of this pathway step by step. Beta oxidation takes place in 4 steps. Each step is catalyzed by a distinct enzyme. So first step is dehydrogenation. In the first step, acyl-CoA is oxidized by the enzyme called acyl-CoA dehydrogenase. A double bond is formed between the alpha and beta carbons of the acyl-CoA chain, entering the beta oxidation cycle. The end product of this reaction is trans-delta 2-enoyl-CoA. This step uses FAD, and this one gets reduced into FADH2. The FADH2 will enter the citric acid cycle and form ATP to be used as energy. The second step is hydration. In the second step, the double bond between alpha and beta carbon of trans-delta 2-enoyl-CoA is hydrated. Name of this product is L-beta hydroxyacyl-CoA. This compound has a hydroxyl group attached to beta carbon in place of the double bond. This reaction is catalyzed by the enzyme enoyl-CoA hydratase. This step requires water. Third step is the oxidation. In the third step, the hydroxyl group present at beta position of L-beta hydroxyacyl-CoA is oxidized by NAD positive. This reaction is catalyzed by 3-hydroxyacyl-CoA dehydrogenase. The end product is beta-ketoacyl-CoA, and NAD positive is reduced to NADH plus H positive. The NADH will enter the citric acid cycle and produce ATP that will be used as energy. Fourth step is thiolysis. Finally, in the fourth step, beta-ketoacyl-CoA is cleaved by a thiol group of another CoA molecule. The cleavage takes place between alpha and beta carbon of the previous product. The end products are a molecule of acetyl-CoA and an acyl-CoA chain, which is 2 carbons shorter than the original acyl-CoA chain that entered the beta oxidation cycle. This was all. Thanks for watching.